So after I did that lecture on vision, I got 36,000 emails from you, the students, asking, what about the other senses? And because I'm all about representation, I created a new chapter just for you about the other sensory systems. I hope you're ready. First on the docket is audition or hearing. Hearing alerts us to many types of useful information. Fun fact, you're probably using it right now. Isn't that crazy? Uh, so, not to be self important, but maybe you're getting some useful information that might be handy on a quiz or test someday, right? So, auditory signals are sensed as periodic compressions of air, water, or other media, also known sometimes as vibrations. That little wavelength, right? That you may have seen before. So we experience hearing, we experience audition by detecting sound waves. As mentioned, those are periodic compressions of air and water or other media, and they vary in amplitude. So how big these waves are, and frequency, how many we see within a period of time. So just so you have some definitions, amplitude refers to the intensity of a sound wave. Frequency is the number of compressions per second, and that is measured in hertz. This is related to the pitch. So something that is lower has a lower frequency. Something that is higher has a higher frequency, and that would be measured in hertz. Timbre, I know it's pronounced timbre. It's timbre. But if you say timbre, then people will be like, cool. But timbre is the way that's pronounced. That is the tone quality or the tone complexity. So, like, maybe you have two different people hitting the same note, but then you hear somebody play that note on a guitar, and then somebody else plays that note on a violin, another person plays it on a piano. It's the same note, it's the same frequency, but the complexity of that tone is the timbre. So here we can see a comparison. Here we have a lower frequency, and we have a higher frequency here. So if you could look in this period, right, we have less waves in the lower frequency versus the higher frequency. Then we have low amplitude versus high amplitude, which is the peak of these waves, right? The higher amplitude has a higher peak, so it's higher intensity. So interestingly enough, the older you get, the worse your ability to perceive higher frequencies becomes. Part of this is just due to normal aging, but also if you're constantly exposing yourself to loud noises. So if you have your headphones in all the time at the highest frequency, or sorry, at the highest volume, then it can affect your ability to perceive those higher frequencies. When it comes to communication, alterations in pitch, loudness, and timbre are going to affect how something is perceived. So if I say to you, "Hi, how's it going?" versus "Hi, how's it going?" Right? That's communicating something to you. One of those, I probably like you and I'm happy to see you. The other one, I'm probably not very happy to see you. Oh, thank you for giving us an example of timbre, Kat. There we go. There's some pitch there. There was some loudness. There's some timbre. What is he trying to communicate to me? I don't know. I gave him food. You guys got a litter box and he can use it. What do you want? Oh, he wants attention. I'm gonna go take a little quick break to give them some attention, and then I'll record the rest of this lecture. Feel free to take a break yourself. Grab a soda, grab something to drink or snack on. Maybe check out that new show on Netflix. Heard it's pretty good. You know, text a friend. You know, text them about what you've been learning. I'm sure they'd be pretty excited to hear about that. So obviously we can't talk about sound without talking about the ear. We have an outer, middle, and inner ear. When we think about our ears, we're usually thinking about the pinna, which is the flesh and cartilage attached to each side of our head. The purpose of the pinna is it alters the reflection of the sound waves and focuses it into the middle ear from the outer ear, and it also helps us locate the source of a sound. The middle ear contains the tympanic membrane, which vibrates when it is struck by sound. It's also known as the ear drum. There are three very small bones, the malleus, the incus, and the stapes, and they help transform waves into stronger waves to the oval window. So the oval window is a membrane in our inner ear, and that transmits waves through the viscid fluid of the inner ear. So outer ear focuses into the middle ear focuses into the inner ear. The inner ear contains a snail-shaped structure called our cochlea. So if you've ever heard of a cochlear implant for people who have hearing impairments, this is what we're talking about. So there are three fluid-filled tunnels, and there are these little hair cells that are auditory receptors that are between the basilar membrane and the tectorial membrane in the cochlea. So those hairs, when they're displaced by the vibrations of the fluid in the cochlea, they'll excite cells of the auditory nerve by opening ion channels. So it's a long process, but essentially you're trying to get these little hairs vibrating because those hairs vibrating are going to signal to the brain that sound is happening. So right now I'm talking, maybe you're hearing me over the speakers on your phone or in your computer, or maybe you have headphones in. So the sound, the vibrations are entering here. So these are the three bones of the ear. So that sound is amplified. It gets into the cochlea, and there are these little hair cells that we can see here that will vibrate and send signals down to the brain. And each of these just takes the area and blows it up. So these, hairs are very small, but you can see that there are a bunch of them. So by amplifying and focusing the sound, specific hair cells will respond to specific vibrations, which will allow us to perceive sound properly. And here are the hair cells very, very close up. Here's a picture with an electron microscope that is inside of you. Cool, right? Or scary. This could either be awe inspiring or the stuff of nightmares. So there are two different theories of how we perceive pitch. One is place theory, which is the idea that the different hair cells will respond to specific frequencies because they're sensitive to only one frequency of sound. There's also frequency theory, which is the idea that the basilar membrane vibrates in synchrony with the sound and causes those nerve axons to produce action potentials at the same frequency. So either we have hair cells that respond to specific frequencies, or the firing of the neurons at specific frequencies triggers us to later perceive those frequencies because they were firing that way. So our current theory is a hybrid of the two. Low frequency sounds are better explained by the frequency theory because those very slow uh, frequencies, those can be explained by firing. If you have something that's super high frequency, right, those neurons would have to be firing pretty rapidly. High frequency sounds are better explained by place theory, the idea that specific cilia are maybe very responsive to a specific range of frequencies. So our auditory nerve produces a lot of impulses. It can be up to 4,000 per second, which is a lot. So no individual axon is firing that quickly. So what is happening is our auditory cells are precisely timing their responses. We really don't yet understand how we hear higher frequencies just because of how quick those wavelengths are, right? So we really need to kind of try to figure out how, if something's at like 10,000 hertz, how we can perceive it. So here's a illustration of the cochlea. Here at the base, we have our higher frequency perception, and at the apex, we have our perception of lower frequencies. So the primary auditory cortex, area A1, is the destination of most of our information from our auditory systems. It is located in our superior temporal cortex. Each hemisphere receives most of its information from the opposite ear. So if you remember our visual cortices, it's very similar to that. We have our what and our where pathways. Our superior temporal cortex allows us to detect the motion of sound. I'm not sure if I can do this right now. I'm not sure if my recording is set to stereo, but if you're listening on headphones, maybe you'll hear the sound moving. If not, then I just look like an idiot right now moving around my microphone, which is fine. That works too. Uh, but if you have headphones or stereo speakers, then you might have heard the sound moving. And that's because your superior temporal cortex allows you to detect the differences between what you're hearing in your left and right ear and use those differences similar to how our eyes look at the differences between the left and right eye. So area A1 is important for auditory imagery, that like mapping of the world using our hearing. We need experience to develop this properly. So axons that lead from the auditory cortex develop less in people who are deaf since birth. So here we have the path of auditory impulses. Just in case you're wondering what the superior olive is, if you ever eat olives too quickly, sometimes they don't go into your stomach. Sometimes they'll go up and get stuck in your brain. And then neurons will form around them and they'll help you with your hearing. So that's why olives are really good for you. The superior olive is just a bundle of brainstem neurons that are helpful in our processing of auditory information. But here you can see a signal from the left ear. We have our cochlear nucleus. We've already talked about the inferior colliculus and the medial geniculate, and here, right here, is our auditory cortex. So our auditory cortex is not necessary for hearing, but for processing the information. So remember the separation between the actual sensation and the perception of those sensations. It provides what's called a tonotopic map, in which cells in the primary auditory cortex are more responsive to preferred tones. So some cells are going to respond better to complex sounds than pure tones. So a tone is like a beep, like a doo, right? That is a pure tone. But if I were playing that same note on a guitar or a piano, right, then that would be a more complex sound. So damage to A1 doesn't necessarily cause deafness unless damage extends to the subcortical areas. So you might see issues with the auditory processing, but it wouldn't be cortical deafness. So here's our primary auditory cortex, and also our secondary auditory cortex. So as you can see from this map, certain areas respond better to certain sounds. So in these areas, we have high response to the highest piano notes and very high squeaky sounds, so like a dog's chew toy or something like that. So those high responsive areas show us increased sensitivity to higher notes. So areas around the primary auditory cortex exist, and they respond better to changes in sound than to just a prolonged sound that is the same. There are also areas that respond best to auditory objects, so specific things that we're trying to pick up. For example, a cat meowing in the background of a recording, machinery noises, and music. So, as mentioned before, sound localization depends upon comparing the responses between your two ears. There are three cues: the sound shadow, the time of arrival, and the phase difference. What we do is we localize low frequency sound by phase difference and high frequency sound by loudness differences. So we'll see an example of a sound shadow in the next slide. But just so you know, when you are listening to something, the sound, if it's coming from one direction, is going to have trouble hearing your other ear. This is the left ear, this is your right ear, right? Because your face is blocking it. So that sound shadow with frequencies between 2 and 3,000 hertz can help us figure out where a sound is located. The difference in the time of arrival at our two ears is also helpful for helping us localize sounds, right? If it arrives in one ear first, then it's probably coming from that side. If it arrives at both ears around the same time, it might be coming from the front or back, right? Also, the phase difference between the ears provides cues to sound localization with frequencies up to 1,500 hertz. Phase differences are basically how this wavelength is where it is in this phase, right? So if you have one that's here and then another one that's here, that phase difference can help us understand where the sound is. So here we can see both localization and the sound shadow, right? So the sound's going to have a harder time reaching the right ear because it has to pass through the brain or bounce off something and then hit the ear, right? So that extra distance is caused by the sound shadow. The differences in arrival times cues the brain into knowing where the sound is coming from. Then, as mentioned, this is a pair of waves that are in phase, but waves that are out of phase, right? So the wavelength is starting at different times, are going to cue us in to where it's coming from. So here's another illustration of phase differences. So if you notice at these two ears, the wave is at different parts of the phase. So that is how a wave would hit you if it's coming at you from the side, right? Now, if you're hearing it from a different angle, that's going to affect the phase. So based off of where the sound hits you, you're going to be able to determine, okay, well, this phase is not what I'm expecting. So that means that it has to be coming from a different angle. The higher the frequency, the harder it's going to be to use that phase detection. So this is for lower frequency sounds. You may have heard the term tone deafness, which is the impaired detection of frequency changes. Essentially, somebody can't recognize pitch easily. So, about 4% of people experience amusia where they really can't differentiate between different pitches. It's associated with thicker than average mass in the auditory cortex in the right hemisphere, but fewer connections from the auditory cortex to the frontal cortex. Some people are more sensitive to pitch. Some people have what is called absolute or perfect pitch, which is the ability to hear a note and identify it, even if it's not like a note played on the piano or a note sung, somebody could hear the humming of a fan and be able to figure out what notes compose that. There's a genetic predisposition that may contribute to it. But the main determinant is early and extensive musical training. So, if as a child you were exposed to a lot of music, then you're going to form those pathways that make it easier for you to recognize pitches out of the air. It is also much more common among people who speak tonal languages, such as Vietnamese and Mandarin Chinese, because from a early age, you're taught to recognize pitch as part of language comprehension, meaning those pathways that you're using to comprehend language can also be used to perceive the tone in music or notes out in the real world. When it comes to hearing loss, there are two categories. There is conductive or middle ear deafness, and then there is nerve deafness or inner ear deafness. Middle ear deafness occurs if bones of the middle ear fail to transmit sound waves properly to the cochlea. This could be caused by disease, infections, tumors, bone growth, etc. Because when you talk, you're actually not using the bones in your ear. Your If you have a normal cochlea and auditory nerve, so they're not affected by anything, you should still be able to hear your own voice clearly. But sometimes people who have an affected middle ear will hear their voice more loudly, or it might be distorted. It might sound louder in one ear than the other. This can be corrected by surgery or hearing aids that amplify the stimulus. Inner ear deafness results from damage to the cochlea, the hair cells, or the auditory nerve. It can vary in degree, and it can also be confined to one part of the cochlea, meaning that people can only hear certain frequencies. It can be inherited, or it can be caused by prenatal problems or early childhood disorders. Then we have tinnitus, which is a frequent or constant ringing in the ears, and it's experienced by many people with nerve deafness. Sometimes it can occur after damage to the cochlea. So the axons that represent other parts of the body innervate parts of the brain previously responsive to sound. So basically, your pathways are getting mixed up, meaning that what ends up happening is your brain, even though it's not getting actual sound signals, thinks that it's getting sound signals. So you are responding to senses that you're not actually receiving. This is similar to the mechanisms of phantom limb. So you are missing maybe your left arm, but because you're receiving signals from other parts of the brain or from the body to the brain, then you might feel like your left arm is itching, even though it's not itching. So tinnitus is like a phantom limb itch for a specific frequency hearing range. As you become older, brain areas responsible for language comprehension become less active. Many older people actually have a decrease in the inhibitory neurotransmitters in the auditory portions of the brain. So it's harder to suppress irrelevant sounds and attend to the important ones, right? I'm not sure if you notice this. This might ruin my lectures forever, but oftentimes there is a fan occurring in the background. Your brain is able to go, oh, well, there's somebody talking and there's a fan. I'm going to ignore the fan and focus on the voice. Some of you, this entire time, that fan has been driving you crazy. And I would apologize, but also, I'm not going to turn the fan off. It is very warm in my room. I at least turned the air conditioner off for you. So I'm meeting you halfway, okay? Uh, I also turned off my air purifier. So I'm inhaling pet dander, smoky air just to do these lectures. You can deal with a ceiling fan for an hour. Is that okay? Anyway, so you're able to tune things out probably, and as you get older, that becomes more difficult. Your attention improves if you watch the face of the person who's speaking, because it helps your brain focus on what's happening. And also, we learn to read lips over time, so it gives us an additional cue to what's being said. So now we're going to switch to the mechanical senses. The mechanical senses respond to pressure, bending, and other distortions of the receptors. These are things like touch, pain, and other body sensations, such as your vestibular sensation, which detects the position of parts of your body and the movement of your head. Audition is a complex mechanical sense because the hair cells are modified touch receptors. So, our vestibular sensation, first of all, you might be going, wait a second, I learned that there are only five senses. Vestibular, that's not one of them, unless taste has like a term that I'm not familiar with. Yes, there are more than five senses, uh, and now you know that. You were lied to as a child because we were like, we can't teach children the word vestibular, but now as adults you're learning. So welcome to adulthood. The vestibular sense is a system that detects the position and movement of your head. So it directs compensatory movements of the eye and helps you maintain balance. So as you're moving your head around, so just move your head around. You might not notice this because your brain's so good at doing this, but your eyes are going to move as well, especially if you're focused on something in the distance, right? So right now I'm looking at my wall, but moving my head around, and my eyes, oops, my eyes were not paying attention to what was on my desk. But my eyes are moving to stay in line with what I'm focusing on in the distance. So that's part of the vestibular sense. The vestibular organ is in your ear and it's adjacent to your cochlea. Our vestibular organ comprises two otolith organs, the saccule and the ultracle, and three semicircular canals. Otoliths are calcium carbonate particles that push against different hair cells and excite them when the head tilts. So when those particles push against the hair cells, we know that we're tilting a certain way. Semicircular canals are filled with a gel-like substance and the hair cells that are activated when the head moves. So these will trigger action potentials that will travel to the brain stem and cerebellum, telling us, oh, we're on balance or we're off balance. If you've ever looked at a level, you know, the little bubble in the little vial of green liquid, it's kind of like that. If the bubble, just imagine that the glass had a sensor that told you if the bubble was hitting one side or the other. So when that bubble moves and touches one side, you go, oh, we're off balance, right? And then you shift and compensate until the bubble's right in the middle. So here we go, right above the ear, right? We have our semicircular canals, our saccules and ultracols. And here we can see the little otoliths. And if you were to tilt back, right, you feel that way, then they all touch this side. So then you would go, oh, look, I'm tilting back, right? And now you understand the vestibular sensation. Uh, Take a break here if you want. Like, I feel like this is a good place for a break. Do you feel like this is good? No? No break? I'm not going to know if you take a break here or not, because this is you're not talking to Professor Thompson. Hopefully, you haven't been talking to me. That would be a little wild if you were doing that. Just a friendly reminder: this is not a live lecture, so if you've been wondering why I haven't been answering your questions, it's because this is pre-recorded. Sorry if I did not make that clear in the first 5 chapters. Now you know. You can listen to this while in the bathroom if you need to. I'm not gonna see or hear, so do what you need to do. You know, you can listen to this while biking, but I will not respond. But also you can take a break whenever you want. You can eat a cookie. Go eat a cookie. Why not? Then we have somatosensation, which refers to the sensation of the body and its movements. This includes discriminative touch, deep pressure, cold, warmth, pain, itch, tickle, and the position and movement of the joints. Our touch receptors vary. They can be a simple bare neuron ending. They can be a modified dendrite, referred to as a Merkel disc. They can be an elaborated neuron ending, or they can be a bare ending surrounded by non-neural cells that modify the function. This stimulation of any of these cells will open sodium channels to trigger, you guessed it, an action potential. This is, for me personally, just a very disturbing photo. You know that thing where you just casually just like lift up your skin and look under it? Ugh. But yeah, this just shows. So here's a nice little hair follicle right there. Within that initial like layer before you get to the deeper stuff, you have all these different receptors, and those receptors are going to be responsive to different signals. This is one of those times where I wish I were like a educational cartoon so I could have a fun song just about all the somatosensory receptors and their probable functions. I don't. I'm sorry. my guitar's out of tune. It's late. You don't want that right now. But just to kind of give an overview so you have an idea, we have free nerve endings. They can be in any area of the skin. They respond to pain and temperature. We have hair follicle receptors. They are located in areas of the skin, weirdly enough, that are covered with hair. Uh, they respond to movement of the hairs. And then we have Meissner's corpuscles. These are on hairless areas and they respond to movement across the skin. So if you were to just run your finger on your palm, that would be a Meissner's corpuscle. If you have a hairy back of your hand, right, it would be a mix of your hair follicle receptors and Meissner's corpuscles. Then we have the Pacinian corpuscles, which respond to vibration or sudden touch. Then we have Merkel's discs, which respond to static touch. If you've ever run your, I was just going to make a joke about static electricity. It's not that. It was going to be a terrible joke. I'm not going to do that to you. But that's just touch that isn't moving. So just sustained. Then Ruffini's endings, which respond to skin stretch. So maybe you're visiting your family and your aunt like pinches your cheeks, right? Those are Ruffini's endings. So next time she does that, say, "Hey, get hands off my Ruffini's endings." She'll laugh because she has a PhD in biology. And then we have the Krauss and bulbs, which are on mostly hairless areas, and we don't know what they do yet. Yay, science! So maybe in 10 years, your big contribution to the field will be figuring out what those do. Congratulations. So the Pacinian corpuscles are responsive to sudden displacement, so like pressure or high frequency vibrations on the skin. If you are not familiar with high frequency vibrations, there's a common experience that many of us can relate to dealing with vibrations on the skin, which is honestly one of my favorite things to experience. Of course, I'm talking about video games that have a rumble feature. It really puts you in the game. So the Pacinian corpuscles have an onion-like outer structure that resists gradual or constant pressure. Sudden or vibrating stimulus bends those membranes and increases the flow of sodium ions, which, as you know, is going to trigger an action potential. Not an onion. That's a Pacinian corpuscle. Merkle disc are receptors that respond to light touch. So, for example, gentle stroking of the skin. Men and women generally have the same number of Merkle discs, but women tend to have smaller fingers. That results in Merkle discs being compacted into smaller or a smaller area and being more sensitive to feeling the distances between those grooves. Now, you might be wondering, why do we need to be so sensitive to temperature? Well, if we can't sense temperature, we're probably going to hurt ourselves, right? Uh, Not only do we have to respond to environmental changes, so for example, if it's too hot, you sweat to cool yourself down. If it's too cold, you shiver to warm yourself up. But if I'm holding something that's boiling hot, well, I'm going to burn my hands. Or if I decide to pull out some ice cubes to carry to my cup, which is, you know, a mile away, at a certain point, my hands are going to respond to how cold those ice cubes are and go, maybe you shouldn't hold these in your hands. This is starting to hurt. So, cold sensitive neurons respond to drops in temperature. They adapt quickly and show little response to constant cold temperatures. So, a sudden change in temperature, so for example, holding an ice cube, is going to cause those to fire. Being in a cold room isn't going to get make those neurons constantly fire because it's constant. Heat sensitive neurons respond to absolute temperature. So it's not a relative change, it's whatever the temperature actually is. So if you are in a 40-degree room and you're holding something that's 50 degrees, it's not going to feel substantially warmer. So chemicals can stimulate receptors for heat and cold. So for example, capsaicin and menthol. So capsaicin is what is in spicy foods. So spicy foods make your mouth feel like it's on fire because it's actually telling your brain that you are experiencing heat. Menthol is the opposite. It tells your brain that you're experiencing cold. So, tickling is something that science does not fully understand. And you have to imagine that the studies for understanding tickling would be really weird, where you just like bring 30 people into a room and tickle them while hooking them up to like an fMRI and see what happens in the brain. So, we don't really understand what and why of tickling, but you know, it's a thing that happens. We do know that the reason why we can't tickle ourselves is because our brain is expecting a stimulation, a specific stimulation, and also sensing that stimulation, and those two together cause a weaker somatosensory response. So they're basically canceling each other out. So information from our touch receptors in our head enters the central nervous system through our cranial nerves. Information from receptors below the head enters the spinal cord and travel through 31 spinal nerves to the brain. I know it's been a minute, so here's a reminder of what the central nervous system looks like. Up here is the brain. You might remember that from having talked about it a lot. And also here's the spine broken up into the cervical, thoracic, lumbar, sacral, and coccyx. So each spinal nerve has a sensory component and a motor component, and it connects to a limited area of the body. We have what are called dermatomes, which are body areas innervated by a single sensory spinal nerve. Now, sensory information entering the spinal cord travel in a well-defined and distinct pathway. For example, a touch pathway is going to be distinct from a pain pathway. So here we can see the different areas of the body and how they are mapped to different parts of the spine. So various aspects of body sensations remain separate all the way to the cortex. So our somatosensory thalamus sends impulses to different parts of our somatosensory cortex located in the parietal lobe. We have different sub areas of the somatosensory cortex that respond to different areas of the body. If you remember a while ago, we talked about the homunculus in the brain, and sorry, I almost said damage. It's the fancy damage. Oh, did your car get damaged recently? Damage to the somatosensory cortex can result in the impairment of body perceptions. So you might receive the sense, but your ability to perceive it properly might be affected. So pain is great because if there's a harmful stimulus, it directs your attention towards that danger. So let's just say you are doing this lecture and you feel a stinging in your leg, and you look down and oh no, there's a scorpion, right? Uh, sorry if any of you are really afraid of scorpions. I was walking the day. I've never. I was walking the other day. I've never seen a scorpion in real life. Like for a very long time, I did not believe that they were real. I believed that they were just a thing that other people made up, but that they couldn't possibly be real. And I was on a little hike and I just saw one and I was just like, oh, they're real. And I just assumed that it was going to kill me. Just like, yeah, we're real. And now you die. So if that triggered you, I apologize. I'm sensitive to that. But if a scorpion stings you, you want to go, oh, it's on my leg. You roll up your pant leg, or maybe you're not wearing pants and you just look down at your leg, and so you go, oh, there's that thing that's hurting me. Now I can solve the problem by shaking it off my leg or pulling out my AR-15 and shooting it off my leg, as is my American right to do so. Stand your ground. So that is the purpose of pain, to find things that are harming us and be able to do something about it, right? Pain sensation begins with the least specialized of all of our receptors, the bare nerve endings. Some pain receptors also respond to acids, heat, or cold. Axons carrying pain info have little or no myelin, so the impulses travel very slowly because they can't do the saltatory conduction. Although the brain processes pain information very rapidly, so the motor responses are quick. So traveling to the brain is slow, but the response to the pain is quick. Mild pain triggers the mild pain will trigger the release of glutamate in the spinal cord. Stronger pain is going to trigger the release of glutamate and is going to release several neuropeptides, including substance P and CGRP. So that's the calcitonin gene related peptide. So if you remember, glutamate is a neurotransmitter, right? The effect on the synapses and surrounding cells is very quick, but neuropeptides are going to have much more long lasting effects. So when you're dealing with a stronger pain, you don't want to just quickly respond to it. Your body is dealing with something that could be pretty terrible. So those neuropeptides are ensuring that the rest of the body and surrounding cells respond properly and continue to respond. When it comes to touch and pain, touch travels on the ipsilateral side, pain travels on the contralateral side. So let's say I'm, with my girlfriend, which I do have. She's real. She's been very busy, which is why you haven't met her, but she is a very real girlfriend. And she holds my hand, right? So I feel that touch from her. And once again, very real. That touch travels on the right side. So right hand being held travels up the right side of my spine. Now, if she's angry with me and she pulls out a knife and stabs me in my hand, well, I'm gonna feel pain. But you know what? It was my fault for forgetting her birthday. And also, just a reminder, she's very real, not made up, real girlfriend. Her birthday is so that pain is actually going to travel on the left side of my spine, right? And it won't cross over until it gets to, is there maybe a bridge in the brain? Did you say pons? Did you say pons? Pons, yep, the pons. Great. So I know you guys are wondering, what about the pain that I feel in my heart? Oh, it's a sad heart. So, emotional associations of pain. Whenever you're feeling emotional pain, these activate a path that goes through your reticular formation of your medulla. Then it goes to several of your central nuclei in the thalamus, amygdala, which, as you might remember, processes emotional information, the hippocampus, the prefrontal cortex, and the cingulate cortex. When we monitor people's brain activity, we find that hurt feelings activate similar pathways as physical pain. Interestingly enough, being wrong about something, so let's just say you had posted an article saying masks don't do anything to stop coronavirus, and then you watched a lot of articles and you're like, oh, actually, masks do a lot to stop coronavirus, and then you felt bad because you were wrong. Feeling wrong activates those same pathways as physical pain. So, different pathways are going to be responsible for our either physical pain reaction or our emotional pain reaction. What do I do with all this pain that I have, Professor Thompson? All this pain in my heart. Well, you could take it and you can put it in your art. Or you could try opioids. Opioids are a super safe, non addictive drug oh, nope, very addictive, not safe at all. Uh-oh. But opioids work by relieving pain. We have opioid mechanisms that are sensitive to opioid drugs and similar chemicals. They bind to receptors found mostly in your spinal cord and your periaqueductal gray area of the midbrain. And these basically help prevent pain signals from getting to where they need to get. Endorphins are a group of chemicals that attach to the same brain receptors as morphine. There are different endorphins for different types of pain. So, when it comes to pain, there's what's called the gate theory. And this proposes that the spinal cord areas that receive messages from pain receptors also receive input from touch receptors and from axons descending from the brain. So these other areas that provide input can close the gates by releasing endorphins, and they decrease the perception of pain. Not the sensation, but the perception of pain. So non-pain stimuli around it can modify the intensity of the pain. So if you've ever like looked down and noticed that you had like bumped yourself and bruised yourself, right? Uh, Sometimes being aware that you've hurt yourself causes you to feel the pain, right? So our context and other mechanisms in our brain and body can affect our perception of pain. Here we can see how endorphins or other substances could affect the sensation of pain. So here we have an afferent axon. We see this pain signal coming down, but this endorphin or these endorphins are hitting these opiate receptors. And so that could cause an inhibitory effect on this presynaptic neuron, meaning that this signal won't send and the pain sensation won't be felt. So there's an area of the brain called the periaqueductal gray area. It is between the third and fourth ventricles. Now, the what the periaqueductal gray area does is it excites an inhibitory pathway. So basically, it's saying, hey, more inhibition. So these axons that are carrying pain messages get inhibited because the periaqueductal gray is saying, hey, no, slow that down. We don't need that. So that's one way of slowing down or interrupting pain messages. There are other ways of relieving pain. We have placebos. So there are drugs that we think might have an effect, but don't actually have an effect. So it decreases the brain's emotional response to the pain perception, but not the sensation itself. We also have cannabinoids, which are chemicals related to marijuana that block certain kinds of pain. These act mainly in the periphery of the body. And then we also have capsaicin, which produces, as we mentioned before, a temporary burning sensation. But what happens afterwards is a longer period of decreased pain. So next time you are feeling a little depressed, just, eat some spicy food. I don't think that's what the lesson is, but try it out. Couldn't hurt. I mean, it could hurt because you could, destroy your esophagus. Okay. If you haven't learned this yet, do not rely on me for medical advice. You should barely rely on me for psychological advice. Do not rely on me for medical advice. But just try it. Next time you're feeling sad, you get broken up with, just eat some spicy food, see how you feel, and tell me about it. Maybe we're on to something. So we have mechanisms in our body that will increase our sensitivity to pain. So when we have damage or inflamed tissue, these are going to release histamines, nerve growth factor, and other chemicals that increase our the responses of nearby pain receptors. Certain receptors are going to become potentiated after an intense barrage of painful stimuli, which can lead to increased sensitivity or chronic pain later. If you've ever wondered why, if you have allergies, you have an itching sensation. So sometimes when my roommate has a dog, and I forget that I'm a little bit allergic to pets, so I'll, rub my face on him. I'm like, oh, you're so cute. You're such a cute little dog. And then my face and hands will itch. I'm like, what's happening? I'm like, oh yeah, I'm allergic. So allergies and other things can release histamines in the skin, which are going to produce an itching sensation. They activate distinct pathways in our spinal cord, which go to the brain, and these impulses travel slowly along this pathway, so half a meter per second. So that's about, for most of us, 2-ish seconds to get through the entire length of our spine. Pain and itch have an inhibitory relationship, so opiates will increase itchiness while antihistamines will decrease itchiness. Antihistamines, think like Benadryl, right? So if you've ever wondered why people who are addicted to opiates complain of itching all the time, now you know opiates are going to increase the itching sensation but decrease the sensation of pain. Now let's talk about the chemical senses. The first sensory system of the earliest animals was chemical sensitivity. A chemical sense enables small animals to find food, avoid certain kinds of danger, and even locate mates. You don't need Tinder when you've got chemical senses. And that is Professor Thompson dating advice. So you know that you can trust it about 32%. Taste is super important. It is a very simple thing. It tells us, do we swallow something or do we spit it out? That is the most important thing that it does for us. Do you swallow or do you spit? There are certain things that I say in class that I'm like, yeah, that is the thing that I said, and it's right there on the slide. So I have to say it. That's an important thing that you need to know. I'm helping by telling you that there's a difference between the two. And now you know. Take that with you wherever you need it to go. So, Just so you know, we like sweet tastes even in infancy. Sweeter tastes have lots of carbohydrates. We are very much attracted to sweet taste. Things that taste sweet tend to give us the energy that we need. They might not always be good for us, but they do give us energy, and energy does keep us alive. We dislike bitter and sour, but we will accept it in small amounts. Or if you are an elementary school child, you will have as much as possible because it is a cool thing to do. We vary in our like of salty flavors, but we do need salt to survive. Taste refers to the stimulation of the taste buds, which are the receptors on the tongue. Our perception of those tastes is flavor, which is a combination of both taste and smell. Taste and smell axons converge in your endopiriform cortex. So if you've ever wondered why it is that the thing that you think is the most delicious thing in the world to other people is not, well, our perception of flavor is just that. It is a perception. It has nothing to do with the objective taste of something. It's based on how we perceive that taste. Receptors for taste, weirdly enough, are modified skin cells. Taste receptors have excitable membranes that release neurotransmitters to excite neighboring neurons. Taste receptors are replaced every 10 to 14 days. This word, not pronounced how you think it would be. It's pronounced papillae, not papillae. I learned that recently. I've been saying it wrong for a very long time. Papillae. So papillae are structures on the surface of the tongue that contain our taste buds. Each papillae may contain up to 10 or more taste buds, and each taste bud contains approximately 50 receptors. Most taste buds are located along the outside edge of the tongue in humans. So, have you ever been up close and personal with the tongue? Uh, Not as close as we're about to get right here. So, here we go. That is your tongue. We can see taste buds real close up, right? Here are the papillae, and here's a cross section of one super close up. So those are in your mouth doing the delightful job of making your cookies and coffee and whatever it is that you decide to put in there taste real good. I'm also imagining that during this lecture, you've all just snuck into your bathroom and like stared at your tongue in the mirror and just gone, ew, gross. That's in my mouth all the time. Ugh. Or maybe you're a mature adult and like not a child like I am, which is completely fine. But also, if you want to just take a break, go to the mirror, stare at your tongue, and just go, ooh, gross. I will allow you that time. I'm here. I'll wait. Okay, that's enough time. You're enjoying it too much. Come back. We're going to talk about more stuff. So when it comes to Western perceptions of taste, we tend to describe four types of taste: sweet, sour, salty, and bitter. And those are our primary tastes. And we have four receptors for them. Procedures that alter one receptor but not others can be used to identify taste receptors. There are substances that can modify taste. So, miracle berries, miraculin, can cause sour things to taste sweet. Gymnema sylvestre tea binds to your sweet receptors and makes it so that you're less susceptible to the effects of sweet things. So, you know, you eat that cupcake right now and you're like, delicious, so sweet and amazing. Those are the words that you would say. You drink some of this tea and it just doesn't taste as good. It tastes more so like, you know, just a plain bagel, maybe even worse than that, right? Nothing wrong with a plain bagel. I like plain bagels, but compared to like a cupcake, you know what I mean? So because those sweet receptors aren't being activated, Gymnema sylvestre tea is sometimes used to help people with diets because if you drink it, then you're less likely to enjoy eating sweet foods, which means you're less likely to eat sweet foods. When we talk about senses, we should talk about adaptation and cross adaptation. Adaptation refers to reduced perception of a stimuli due to the fatigue of receptors. Cross adaptation refers to reduced response to one stimuli after exposure to another. Some of you may have heard me mention four senses and gone, wait a second. And I did wait a second, and now I'm responding. Even though we talk about sweet, sour, salty, and bitter as the primary senses, there is evidence that suggests a fifth type of glutamate receptor, which is umami, or the savory taste. This is MSG. Just fun fact, some of us may have learned that MSG is bad and terrible, and it has all sorts of terrible effects. First of all, it's natural. It occurs in our body and occurs naturally in food that doesn't have it added. Secondly, it's delicious. Third, the reason why that fear of MSG, especially in Chinese food, resulted was because, and this might surprise you, that there was something racist that happened in America, but it was because of racism. Basically, in order to like discredit the deliciousness that is Chinese food, people started talking about how they would have Chinese food dreams and blah 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 and all these side effects. And a article got published. It got very popular, and since then, people have had this fear of MSG and also of Chinese food. But all of that is unfounded. All of that is very racist. So next time somebody's complaining about MSG, just say, get out of here with that, because it's not founded in science. So, and now you can have all the MSG that you want and just enjoy it and not have to worry about it. So there's also some research that suggests that there's an ability to taste fats, and that would be a sixth type of taste. For now, that's being called oleogestis. Oleogestis also sounds like a forgotten Roman emperor. All hail Caesar Oleogustus the Third. That's that's the best joke I can come up with for taste perception. You're getting my best humor, so you're welcome. Here you can see the responses in a rat brain to four different tastes. So you have on the top sucrose, which is sweet. Then you have the N for NaCl, table salt, which, as you might guess, is salty. Then you have hydrochloric acid, which is, from what I've told, very sour. And then you have quinine, which is bitter. So you can see those different responses based off of the type of sense. So let's talk about the mechanisms of these taste receptors. Saltiness receptors permit sodium ions to cross the membrane that results in an action potential. Sour receptors detect the presence of acids. sweetness, bitterness, and umami receptors activate a G protein that releases a second messenger in the cell when a molecule binds to a receptor. So we have a sensitivity to bitter taste because many things that are bitter are actually toxic. About 25 types of bitter receptors are sensitive to a wide range of chemicals with varying degrees of toxicity. Most taste cells contain only a small number of these receptors, but we are sensitive to a wide range of harmful substances, but not highly sensitive to any single one. So when it comes to something that can poison us, we don't really need to figure out what type of poison it is. We taste it, we go, oh no, this is not good. And then we spit it out, right? So that's why we don't really need to discriminate between the different ones. We just need to know that it is one of those, and then we deal with it. Different nerves are going to carry taste information to the brain from the anterior two-thirds of the tongue rather than from the posterior tongue and throat. So this is your tongue, right? So about here is where most of the information is being carried. And this makes sense, right? By the time it gets to the back of the tongue, you're already preparing to swallow it. Most of the time, you don't put things right on the back of your tongue, so it makes sense that the beginning part is more sensitive and taking more of the information back. Taste nerves project to a structure in the medulla known as the nucleus of the tractus solitarius, the NTS. This projects information to various parts of the brain. Like our other senses, various areas of the brain are responsible for processing different taste information. The somatosensory cortex is going to respond to the touch aspect of taste, and the insula is the primary taste cortex. Each hemisphere of the cortex is also responsive to the ipsilateral side of the tongue. So you have your tongue here, you have your brain here, left tongue, left brain, right tongue, right brain. So our thalamus and cerebral cortex receive impulses from both the left and right sides of the tongue. And here you can see the insula, or our primary taste cortex. So differences in how we perceive taste can be related to a lot of different things. One, we have taste sensitivity, which is going to be related to the number of fungiform papilla near the tip of the tongue. Genetic factors and hormones can also affect this. So we have people who are super tasters who have a higher sensitivity to all tastes and mouth sensations in general. Also, if you are pregnant, you are more likely to have a higher sensitivity to taste. People who have low sensitivity to taste are called non-tasters, very creative name. And you can compare populations in the US versus, let's just look at Japan, right? So there's a pretty significant difference in if you're comparing like 11% to 28%, the number of people who don't have increased taste sensitivity. So if you look at the differences between a super taster and a non-taster, there are densely packed fungiform papilla in the super taster as compared to the non-taster. So if you want to uh, check to see if you're a super taster, a taster, or a non-taster. You can do this experiment. So feel free to, if you just happen to have a quarter-inch hole punch, small piece of wax paper, cotton swab, blue food coloring, flashlight, and magnifying glass, feel free to just, pop this out real quick. Olfaction is our sense of smell, the detection and recognition of chemicals that contacts the membranes inside of our nose makes up olfaction. So this helps us find food, it helps us find mates, and it helps us avoid danger. Rats and mice show an immediate unlearned avoidance of the smells of cats, foxes, and other predators. So if, you had some cat smell and you put it on, one piece of cloth, and then you had a, very sexy rat smell, and you put it on another piece of cloth, even if the uh, rat had never seen a cat before, right? There is an inborn response where the rat is going to avoid that smell. But if you didn't have the ability to smell, you wouldn't get that signal saying, oh, this smell is scary to me. And then I'm just going to assume that these two became best friends. We can only hope. Even though we tend to think that following a scent is an animal thing to do, we can actually learn to do this. We won't be as good as, say, like a bloodhound, but we can get better with practice. Scent is important for our food selection. A lot of what we perceive as taste is actually olfaction. So if you are really congested, or if you just plugged your nose while you were eating, you'd notice that a lot of your perception of taste would be affected. So scientists can have fun. They found humans who were willing to try to follow a scent, and many humans are able to do it pretty accurately. So, in case you just want to become a super smeller at some point and really hone your nose, you can do it. I believe in you. When it comes to social behavior, we tend to prefer the smell of potential romantic partners who smell different from ourselves and our family members. This decreases our risk of incest, which genetically is pretty bad because it increases your chances of uh, building immunity and uh, not having sex with family members increases your chances of having children who are going to survive longer because any genetic problems are less likely to be handed down. Our olfactory cells line the olfactory epithelium in the rear of our nasal passages and are responsible for smell. Olfactory receptors are located on the cilia, which extend from the cell body into the mucous surface of the nasal passage. We have hundreds of olfactory receptors. By we, I mean vertebrates, and they are highly responsive to some chemicals but unresponsive to others. So, if you are getting your COVID test, right, your swab goes up here, and up in this area where our uh, olfactory receptors are. Here's a nice little close up of them. Proteins in olfactory receptors respond to chemicals outside of the cells and trigger changes in the G protein inside the cell. That G protein triggers chemical activities that lead to what? An action potential. And here is a olfactory receptor protein. So when it comes to the brain, axons from our olfactory receptors carry information to the olfactory bulb. Chemicals that smell similar excite neighboring areas. Chemicals that smell different excite more separated areas. Coding in the brain is determined by which part of the olfactory bulb is excited. The olfactory bulb sends axons to the cerebral cortex, where messages are coded by location. Sometimes our olfactory receptors are damaged, so they're replaced approximately every month because they are vulnerable to damage from contact with the air, and they are also subject to permanent impairment from massive damage. So let's just say there was, you were just constantly, I don't know, snorting cocaine, right? You could permanently affect your ability to smell. As you are already aware, individual differences in olfaction do exist. Women do detect odor more readily than men, and the ability to detect a faint odor and become more sensitive to it is characteristic of young adult women. It seems to be influenced by hormones. Mice with a gene that controls a channel through which most potassium travels to reach the olfactory bulb developed a sense of super smell. You may have heard of pheromones, the chemical that makes you attractive. That is defined as a chemical released by an animal to affect the behavior of others of the same species. So usually it's discussed in relationship to sexual behaviors, but it's not necessarily always sex. The vomeronasal organ, the VNO, is a set of receptors found in most mammals located near the olfactory receptors that are sensitive to pheromones. The VNO and pheromones are important for most mammals, but less so for humans because we don't always choose our mates based off of how they smell, even though them smelling nice is nice. The VNO is very tiny in human adults and has no receptors, so it's considered vestigial. So it is a thing that is an evolutionary remainder of something that we used to have, but doesn't serve much of a purpose anymore. We unconsciously respond to some pheromones through receptors in the olfactory mucosa. So, for example, sometimes women can synchronize their menstrual cycles, and this can happen unconsciously through smell. Last but not least, let's talk about synesthesia. Synesthesia is the experience of one sense in response to stimulation of a different sense. So some people see numbers or letters as a specific color. This tends to cluster in families that also have perfect pitch. So there seems to be a genetic predisposition for this mixing of senses. Synesthesia is actually pretty cool. That sounds so cool saying that. Synesthesia is pretty cool. I just adjusted my glasses. Hope you heard that. The But if you are interested in like cool, fun, weird things that our brain does, I would say check out synesthesia because the different types of synesthesia that people can have do vary. There are few major categories, but it's pretty interesting to see how these things happen. So feel free to get on the YouTube, which you already are on, and check it out. See what you can find about synesthesia, and yeah, that's it. That's the end of the chapter. We did it. I'm really proud of us.